So now we're talking about palynology evidence. This is one of the my favorite topic. I have so much interest in palynology once, but unfortunately I changed my direction in research and studies. Okay, so when I talk about palynology, it is the analysis of pollen grains and spores as a method of taxonomical characterization. So this is represent as part of palynology. So palynology can be translated literally as the science of floating dust particles in the air. In this case, dust refers to the pollen. So the pollen wall has been a subject of considerable attention, especially in an attempt to establish the evolutionary history of angiosperm. So talking the pollen, especially the pollen wall, is used for looking at the angiosperm history in terms of evolutionary. So spore is a haploid cell. So this is derived from the sporangium via meiosis, whereas pollen is an immature endosporic male gametophyte derived from male spores. So sometimes we call it as a microspores in seed plants. So some families such as Asteraceae, okay, show different types of pollen. So like you can look at Eurypalinus. So whereas several others have single morphological pollen types of Stena palinus. So certain families has only one types of pollen. We call it as Eurypalinus, and if we have several types of pollen in one plant, we call it as a Stena palinus. Okay. And such Stenopalinus groups are considerable significance in systematic palynology. So a lot of research on this Stenopalinus. So pollen grain also present a number of features of taxonomy's interest. The number of nuclei present at the time of shedding is also significant. Most primitive angiosperm are shed at 2 nucleate stage, whereas in more advanced groups, pollen is shed at 3 nucleate stage. So other parts, also angiosperm, mostly have pollen grains of radial symmetry, bilateral symmetry being found in several gymnosperm. So you can look at different. So radial symmetry, angiosperm as basically radial symmetry. Gymnosperm are bilateral symmetry. So you can look when you can look at this to differentiate of symmetry of the pollen, you can say that you can differentiate between angiosperm and gymnosperm in the preliminary stage. So most pollen grains are globous in shape, although boat-shaped, ellipsoidal, and fusiforms are also met in different angiosperms. So since most pollen grains, at least in early stages, form tetrads, okay, the outer end of grain is termed distal pole, whereas the inner end where grains meet as we call it as a proximal pole. We have a distal pole and also proximal pole, and the line joining the two poles we call it as a polar axis. Okay, uh, and then the line running around the pollen at right angles to the polar axis is termed the equator. Okay, so you can look at this, the part of the pollen grains. So all these uh, characteristics of polar, okay, characteristics of the polarization of the pollen can be used for taxonomic differentiation of between plant species. Okay, you can pause this video to look at these figures. Okay, and when you talk about pollen in terms of characteristic, one of the major use of pollen for taxonomic evidence is the aggregation of the pollen itself. So Microsporogenesis, the process of the microsporogenesis, can yield four microspores, which measure into pollen grains. So, in large majority of angiosperm, the pollen grains separate prior to release. So we have such single pollen grains we call as monads. In some rare cases, we have pollen grains released and fused in pairs. We call it as dyads. In many angiosperms, so most of the angiosperm, we have the four microspore do not separate, and the pollen grains form what we call as tetrad. So these are the formation: monad, dyad, and tetrad. So in some genera, such as the Coleandra here, the pollen grains are connate in a group of more than four. So such pollen grain constitute we call it as polyads. Polyad. The polyad generally consists of 8 pollen grains. Okay, 8 pollen grains. So 2, 4, 6, 8 here. But can but rarely in can be more than 10. Okay, so around 8 and 10 here. So in subfamily Acilipedidae of the family Apocynaceae and several members of Orchidaceae, we have what we call as all pollen grains of a tica are fused into a single mass known as pollinium. So in general, in the plural is a pollinia. Okay, so this is how the pollen aggregation looks like. Of course, sorry. Of course, when you look at pollen aggregation, you can look at the important part to in angiosperms is how the formation of these four tetrads. So we have different types of the four. So the most common one is a tetrahedral tetrad, then linear tetrad, rhomboidal tetrad, tetragonal tetrad, and also decussate tetrad. Okay, so these are the most common one in the angiosperms. So at the same time, the pollen grain wall also is made of two principal layers. So these are also can be used uh, for for identification or classification of the plant. So using the pollen, so you have the axine and also the intine. So the axine is a hard and impregnated with a sporopollenin, a substance that make it resistant to decay and enables preservation in fossil records. So axine is further differentiated into two layers we call as the outer ectacine and inner endexine. So the ectacine is further distinguished into basal, foot layer, radially elongated columella, and roof-like tectum. Okay. Of course, above layers of axine are clearly visible. Uh, especially under an electron microscope, but when observed under a light microscope, the inner layer known as an nexine includes endexine plus foot layer of ectaxine. So the upper layer is consisting of columella, tectum, and the supratectal sculpturing constitutes axine. So of course, a lot of terminology that I used here. I'm not. I don't want you to become overwhelmed with all this. Uh, overwhelmed with the terminologies. I'm just saying this so you can become familiarized. When you go into details to study the pollen wall later on in your life in your study, you can revisit and then look at these terminologies again. Of course, when you talk about the pollen wall sculpturing, when you have the pollen wall, we have the how it's sculpture. So within the pollen morphological main groups or classes, the differences in the structure and also the sculpturing of the axine of the pollen grains are important characteristics for identification. So majorly, we have the characters of the pollen wall. So basically, for identification, we use the sculpture of the pollen wall. So the inner structure of the axine can be either tectate or intectate. So the sculpturing of the pollen grain is the ornamentation of the axine surface, which can be ciliate, foveolate, areolate, gamete, clavate, verrucate, baculate, acinate, regulate, striate, or reticulate. So these are all the structures that you can see. This is in the 3D and it is in a 2D. So spores basically do not have a characteristic structuring of the cell walls. So overview of the woody plants, terrestrial forbs, and aquatic plants represented in the pollen morphological classes, like in what presented here. Finally, the one that mostly used when we talk about this one is talking about pollen aperture. So this is a specialized region of pollen wall through which the pollen tube comes out. Okay, so an aperture is a thin or missing part of the axine, which is independent of the patterning of the axine. So two different types of apertures can be distinguished, which is the pores and also the fissures. It's easy for me to call it colpi. The latter are more primitive. They are elongated with pointed ends. Pores are usually isodiametric. They can also be slightly elongated, but in contrast to colpi, they have rounded ends. So in some pollen grains, the the axine around the apertures is either thicker or thinner. In pores, this border is termed annulus. Basically, this is a typical in the grass pollen and in colpi and in colpi margo. So, for instance, in the Hedera helix. Pollen grains with spores are porate and those with colpi are colpate. If both spore and colpus are combined in the same aperture, the pollen grain is called colporate. So again, please refer to the textbook for more details on the plant families according to the plant aperture. So not many experts in the country right now for talking about palynology. I hope some of you can venture in this palynology field. If you venture, I want to become your student later on. This is an exciting field for sure.